Thanks.
Time as any, Mr. Newman. Take her down to periscope depth.
Yes, sir. Clear the bridge. Dive.
Stand right.
Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova, and today in this episode of Nerds Go Clubbing, we'll be covering the article Genomics and the Irreducible Nature of Eukaryote Cells. The term clubbing is a play on words. In the United States, we have people that like to go to nightclubs, and that's called clubbing. In the case of researchers, science researchers, they frequently have informal gatherings that they call journal clubs, where they'll take a scientific article and they'll read through it together. So that's what we're going to go through today. And this is the article here. The Irreducible Genomics and the Irreducible Nature of Eukaryote Cells by Kurland, Collins, and Penny. So this is part two. In part one, we just covered like the first page, and it was just kind of the basics, going through what made eukaryotes different from prokaryotes. Prokaryotes include things like bacteria and archaea. And one interesting thing that they mentioned is here. Let me see if I can make this bigger so you all can see it. Is the common ancestor of eukaryotes, bacteria, and archaea may have been a community of organisms. So that's kind of interesting that they're suggesting that there wasn't one single original cell for the common ancestor eukaryotes, bacteria, and archaea. That raises the question whether there was a single cell at all at the beginning of all life. And we don't need to go there today. I just thought that was interesting. So the beginning of the article, I'm highlighting it here. Hopefully you could see my cursor. It described the parts that made, that were salient characteristics of what constituted a eukaryote. things like nuclei, nucleoli, Golgi apparatus, centrioles, and endoplasmic reticulum. And it said that they are examples of cellular signature structures. The acronym is CSSs, that distinguish eukaryote cells from archaea and bacteria. And they also coined another acronym, ESP, Eukaryote Signature Proteins. So it says here, these are hundreds of proteins with no orthologs evident in the genomes of prokaryotes. That's just, that's a fancy way of saying that these are proteins that are only in eukaryotes, not in prokaryotes. So just remember that. And we may just begin where we left off with that introduction. They mentioned, interestingly, that even the mitochondria, which is considered something of a prokaryote that was engulfed or absorbed into this eukaryote cell, since so many of the genes in the mitochondria are actually in the nucleus of the eukaryote, they consider the mitochondria as, and you'll see the word here, as an honorary CSS, cellular signature structure. So now we'll continue where we left off from part 1. There are substantial numbers of ESPs, that is eukaryote signature proteins, in the other CSSs, that is cellular signature structures. For the proteome of the reduced anaerobic parasite Giardia lamblia, which is a eukaryote, searches of 2,136 proteins found in each of the Saccharomyces cerevisiae yeast, Drosophila melanogaster, which is a fruit fly, Canorhabditis elegans, which I believe is a worm, the nematode, and Arabidopsis thaliana, which is a plant, yielded 347 ESPs, eukaryote signature proteins, for G. lamblia. This was reduced to roughly 300 by rigorous screening, with ESPs distributed between nuclear and cytoplasmic compartments. And that was referring to figure 2. So they're proteins that are, I guess, are distributed in the nucleus, and proteins distributed in the cytoplasm. The ubiquity of the ESPs and the absence of archaeal descendants are not easily explained by a prokaryote genome fusion model. Wow. The ubiquity of ESPs and the absence of archaeal descendants are not easily explained by a prokaryote genome fusion model. The simplest interpretation is that the host for the endosymbiont mitochondrial lineage was an ancestral eukaryote. The simplest interpretation is that the host for the endosymbiont mitochondrial lineage was an ancestral eukaryote. Let me think on that. That's suggesting that that there was an ancestral eukaryote that then had an endosymbiosis event, which is interesting because since the mitochondria is the powerhouse of the cell, there had to be a way for this ancestral eukaryote to be able to generate ATP. And maybe they're not going to cover that. I think no one would know how that would work exactly, even though there might be biochemical pathways where ATP can be generated outside of the ATP synthase in a mitochondria. It still seems kind of problematic to me. Anyway, similar results are obtained for another reduced eukaryote, the intracellular parasite Encephalitozoon cuniculi. E. cuniculi. A recent study identified 401 ESPs, of which 295 had homologs among the ESPs of G. lamblia. Two major categories of ESPs in the G. lamblia and E. cuniculi genomes were distinguished: those associated with the CSSs, which is the cellular signature structures, and those involved in control functions such as guanosine triphosphate binding proteins, kinases, and phosphatases. It was also observed that many characteristic eukaryotic proteins, I lost my place. Apologies. It was also observed that many characteristic eukaryotic proteins with weak sequence homology to prokaryotic proteins, but more convincing homologies of structural folds such as the actins, tubulins, kinesins, ubiquitins, and some GTP binding proteins are among the most highly conserved eukaryotic proteins. These may be descendants of the common ancestor recruited early in the evolution of the eukaryotic nuclear genome. Nucleolar proteomes are examples of essential eukaryote compartments not wrapped in double membranes and where there is no suspicion of an endosymbiotic origin. From 271 proteins in the human nucleolar proteome, 206 protein folds were identified and classified phylogenetically. Of these, 109 are eukaryotic signature folds, and the remaining ones appear to be descendants of the common ancestor, occurring in 2 or 3 domains. The spliceosome is a unique molecular machine that removes introns from eukaryote mRNAs. Even though we do not know the ancestral processing signals for the earliest eukaryotes, roughly half of the 78 spliceosomal proteins likely to be present in the ancestral spliceosome are ESPs, that's eukaryote signature proteins, whereas the other half, containing the SM-LSM proteins, have homologs in bacteria and archaea. These distributions of both ESPs, as well as of putative descendants of the common ancestor, suggest that many components of the modern spliceosomes were present in the common ancestor. The subdivision into subcellular compartments, CSSs, with characteristic proteomes restricts proteins to volumes considerably smaller than the whole cell. The subdivision into subcellular compartments, CSSs, these are cellular signature structures, with characteristic proteomes restricts proteins to volumes considerably smaller than the whole cell. Concentrations of macromolecules in cells are very high, typically between 20 and 30% of weight or volume. Such densities are described as molecular crowding because the space between macromolecules is much less than their diameter. Consequently, diffusion of proteins in cells is retarded. Molecular crowding favors macromolecular associations, large complexes, and networks of proteins that support biological functions. High densities enhance the association kinetics of small molecules with proteins because the excluded volumes of the proteins reduce the effective volume through which small molecules diffuse. The sum of these effects is that the high macromolecular densities within CSSs enhance the kinetic efficiencies of proteins. The same principle applies to smaller prokaryotic cells, but the effects are accentuated in larger cells. Subdividing high densities of proteins into more or less distinct compartments containing functionally interactive macromolecules is expected to be an early feature of the eukaryote lineage. The distinctive proteome of nucleoli demonstrates that compartmentation does not require an enclosing membrane. The distinctive proteome of nucleoli demonstrate that compartmentation does not require an enclosing membrane. And let me go over here. I presume that they're talking about this, the nucleoli, and it doesn't need compartmentation. Wow. Furthermore, cell fusion is not required to account for, nor does it explain the large number of eukaryote cell compartments. So I think cell fusion is referring to endosymbiosis. It's not needed. One doesn't need to assume endosymbiosis. That's the way I'm reading it anyway. To account for or explain the large number of eukaryote cell compartments. So yet another thing endosymbiosis doesn't solve. Selection gives and selection takes away. Well, and selection takes. Genomes evolve continuously through the interplay of unceasing mutation, unremitting competition, and ever-changing environments. Both sequence loss and sequence gain can result. In general, expanded genome size, along with augmented gene expression, increases the costs of cell propagation. So the evolution of larger genomes and larger cells requires gains in fitness that compensate. Conversely, genome reduction is expected to lower the costs of propagation. There is an ever-present potential to improve the efficiency of cell propagation by reductive evolution. There is an ever-present potential to improve the efficiency of cell propagation by reductive evolution, which, by the way, I should mention in passing, that is akin to genetic entropy, where natural selection helps destroy function. Anyway, environmental shifts may neutralize sequences, leaving no selective pressure to maintain them against the persistent flux of deleterious mutations. Such neutralized sequences eventually and inevitably disappear because of mutational meltdown. Oh boy, that's another reference to what creationists call genetic entropy. So reductive evolution, mutational meltdown are instances of genetic entropy. Genome reduction can be achieved through differential loss of coding and non-coding sequences. Compaction. So let me try to read through this again. Differential loss of coding and non-coding sequences. Compaction. Theileria has evolved through gene loss as well as compaction of its intergenic spaces, whereas paramecium has eliminated only a small length of genes, but markedly reduced the number of its introns. The complex genomes of some vertebrates, pufferfish, tachyfugu, are so highly compacted that their genome lengths are reduced to one-eighth that of other vertebrates. Their genome lengths are reduced to one-eighth that of other vertebrates. Extreme cellular simplification is observed among anaerobic protists, including simplification of CSSs, that is, cellular signature structures, such as mitochondria and the Golgi apparatus. S. cerevisiae, yeast, which underwent a whole genome duplication, subsequently purged 85% of the duplicated sequences. The evolution of genome content is clearly not monotonic. Genome sizes on the branches of a phylogenetic tree of fungi show irregular genome enlargement, including duplication and reduction. Examples of ecological circumstances driving genome reduction are seen in many intracellular endosymbionts and parasites, which gain few genes but lose many genes responsible for metabolic flexibility. Wow, that can't be good. I don't know how easy it is for eukaryotes to do horizontal gene transfer. So if they're losing genes, that can't be good. The mitochondrion is even more extreme in its reductive evolution. Its ancestral bacterial genome has been reduced to a vestigial microgenome supported by a predominantly eukaryote proteome. That's interesting. Genomes of modern mitochondria encode between 3 and 67 proteins, whereas the smallest known free-living alpha protobacterium, Bartonella quintana, encodes 1,100 proteins. Taking Bartonella as a minimal genome for the free-living ancestor of mitochondria, nearly all of the bacterial coding sequence have been lost from the organelle, though not necessarily from the eukaryote cell. The mitochondrial genome of the proteus Reclinomonas americana is the largest known, but has still lost more than 95% of its original coding capacity. Wow. This abbreviated account of genome reduction illustrates the Darwinian view of evolution as a reversible process in the sense that eyes can be acquired and eyes can be lost. Genome evolution is a two-way street. This bidirectional sense of reversibility is important as an alternative to the view of evolution as a rigidly monotonic progression from simple to more complex states, a view with roots in the 18th century theory of orthogenesis. Unfortunately, such a model has been tacitly favored by molecular biologists who appear to view evolution as an irreversible march from simple prokaryotes to complex eukaryotes, from unicellular to multicellular. There are many well-documented instances of genome reduction. The many well-documented instances of genome reduction provide a necessary corrective measure to the often unstated assumption that eukaryotes must have originated from prokaryotes. Let's read that again. That seemed kind of important. The many well-documented instances of genome reduction provide a necessary corrective measure to the often unstated assumption that eukaryotes must have originated from prokaryotes. Whoa, are they sort of suggesting eukaryotes didn't descend from prokaryotes, that they're just going to leave it kind of ambiguous? The hunt for the phagotrophic unicellular raptor. The hunt for the phagotrophic unicellular raptor. Proteomics, together with comparative genomics, allows glimpses of the cell structure of eukaryote ancestors. They are likely to have had introns as well as the complex machinery for removing them. And much of that RNA processing machinery still exists in their descendants. Because of molecular crowding, it is expected that interacting proteins would tend to accumulate in functional domains, making rudimentary CSSs, that is, cellular signature structures, early features of the large-celled eukaryotes. We cannot say whether there was a substantial period of time. We cannot say whether there was a substantial period of time after the emergence of cells when there were no unicellular raptors or predators, a Garden of Eden. However, the identification among prokaryotes of orthologs as structural affinities to actins, tubulins, kinesins, and ubiquitins is consistent with some early organisms having evolved a phagotrophic lifestyle. This echoes a recurrent theme in which it was supposed that the earliest eukaryotes could feed as unicellular raptors. So let me look up that word, phagotrophic. Phagotrophic, feeding by engulfing a food cell or particle and ingesting it in a phagocytic vacuole in a manner of some flagellates. So this is like a cell that eats other cells. Cool. I call them raptors. I like that. Unicellular raptors. We expect that the earliest organisms were primarily auxotrophs, heterotrophs, and saprotrophs. We covered that in part 1. It's still kind of hard to figure out for the uninitiated like me what those are. An excellent community to support raptors. Phagotrophy is a hallmark of eukaryotic cells. I didn't know that. Phagotrophy is a hallmark of eukaryotic cells and is unknown among modern prokaryotes. I didn't know that. I did not know that. And so it is natural to reconsider this feeding mode as a defining feature of ancestral eukaryotes. Cavalier-Smith suggested that the ancestors of eukaryotes were phagotrophic, anaerobic, free-living protists called archaeozoa. He also identified present-day anaerobic parasites such as entamoeba, giardia, and microsporidia as archaeozoa. However, these organisms are descendants of aerobic mitochondriate eukaryotes. Genome reduction and cellular simplification are hallmarks of parasites and symbionts. Genome reduction and cellular simplification are hallmarks of parasites and symbionts. Indeed, most of the eukaryotic anaerobes studied so far are parasites or symbionts of multicellular creatures. For the reasons outlined above, we favor the idea that the host that acquired the mitochondrial endosymbiont was a unicellular eukaryote predator, a raptor. Whoa. So I think I saw this debate in a later paper that Emery Moyna and I were talking about regarding the lack of a LECA, last eukaryotic common ancestor. And in that, there was a debate whether the eukaryote existed before it acquired a mitochondria, whether the eukaryote existed before it acquired the mitochondria or after. So, of course, the creationists just believe these were all created. Not to mention that the mentions of reductive evolution would suggest that the natural direction of evolution is from a created complex state that's under continuous reduction. And the general trend is loss of complexity. Anyway, the emergence of unicellular raptors would have had a major ecological impact on the evolution of gentler descendants of the common ancestor. These may have responded with several adaptive strategies. They might outproduce the raptors by rapid growth or hide from raptors by adapting to extreme environments. Thus, the hypothetical eukaryote raptors may have driven the evolution of their autotrophic, heterotrophic, and saprotrophic cousins in a reductive mode that put a premium on the relatively fast-growing streamlined cell types we call prokaryotes. Thus, the hypothetical eukaryote raptors may have driven the evolution of their autotrophic, heterotrophic, and saprotrophic cousins in a reductive mode that put a premium on the relatively fast-growing streamlined cell types we call prokaryotes. And I don't know when they referred to this figure here. Figure 3, genome sizes in megabases can increase and decrease in lineages because of events such as gene duplication and reductive evolution, as illustrated in the fungal phylogeny. Genome sizes were obtained from the National Center for Biotechnology Information, the NCBI, genome biology database, GD, genome duplication, and RE is reductive evolution. Okay, so it's just showing here this phylogenetic tree where there's gene duplication and then reductive evolution. Okay, so now we're at concluding remarks. Genomics and proteomics have greatly increased our awareness of the uniqueness of eukaryote cells. This, together with increased understanding of molecular crowding, as well as the dynamic, often reductive nature of genome evolution, offers a new view of the origin of eukaryote cells. The eukaryotic CSSs, that's cellular signature structures, define a unique cell type that cannot be deconstructed into features inherited directly from archaea and bacteria. Whoa, okay. This is suggesting an independent origin of some sorts. Let me just read that again. The eukaryotic CSSs define a unique cell type that cannot be deconstructed into features inherited directly from archaea and bacteria. And God willing, this Sunday, Dr. James Carter and I will actually be talking about some of this, that even for the homologs, for the homologous proteins, these are features that can't be directly inherited because of the problem of intercellular trafficking. We'll cover that. I mean, this is true. This problem that the authors point out is true on so many levels. So let me read it one more time. The eukaryotic CSSs define a unique cell type that cannot be deconstructed into features inherited directly from archaea and bacteria. Only a small fraction, roughly 15%, of alpha proteobacterial proteins are identified in the yeast and human mitochondrial proteomes. None seem to be direct descendants of archaea, and roughly half seem to be exclusively eukaryotic. Whoa. The identification of alpha proteobacterial descendants in this proteome validates the phylogenetic distinction between direct descent from genes transferred to the host from the bacterial endosymbiont as opposed to descent from a hypothetical common ancestor. The identification of proteobacterial descendants in this proteome validates the phylogenetic distinction between direct descent from genes transferred to the host from the bacterial endosymbiont as opposed to descent from a hypothetical common ancestor. ESPs, that is, eukaryote signature proteins, are important markers of the novel evolutionary trajectory of modern eukaryotes. In contrast, most proteins occur in more than one domain, and most of these could derive from the common ancestor. We take the relative abundance of signature proteins among eukaryotes to indicate their genomes typically have a greater coding capacity than those of prokaryotes. It remains to be seen which ESPs have been lost from prokaryotes and which have been acquired by eukaryotes during their evolution. The hypothetical fusion of an archaeon and a bacterium explains nothing. Let me say that again. The hypothetical fusion of an archaeon and a bacterium explains nothing about the special features of the modern eukaryote cell. Let me read that again. The hypothetical fusion of an archaeon and a bacterium explains nothing about the special features of the modern eukaryote cell, nor the many signature proteins. Nothing in global phylogenies based on ribosomal RNA, pooled proteins, and protein fold families indicates that genome fusion generated the eukaryote lineage. Nothing in global phylogeny. Nothing in global phylogeny. I'm going to repeat that. Nothing in global phylogeny indicates that genome fusion generated the eukaryote lineage. So there again, I'm just trying to emphasize that phylogeny doesn't explain squat. It's an assumption. It's not a mechanistic explanation for why things actually work. New machines emerge. All you're just saying is we're just assuming these are related because we have some proteins that are similar, but it doesn't explain the origin of the other proteins, except to say, oh, well, they're just related. We don't really know how they came about. And so again, phylogenies don't explain anything. So if one assumes universal common ancestry and that hypothetically the phylogeny does trace the ancestral lineage, it's still not a mechanistic explanation for how these features emerged. I keep trying to hammer evolutionary biologists that phylogenetic reconstructions are not mechanistic explanations. What I mean by mechanistic explanations is why physics and chemistry make the emergence of de novo systems like these cellular signature structures, why it makes it highly probable. In fact, everything we know about chemistry, physics, and engineering principles and probability says that these shouldn't be highly probable because of all the coordinated complexity in these systems. So let's continue. Perhaps interest in fusion models arose because BLAST searches suggest that different eukaryotic coding sequences are sometimes more closely related to archaeal homologs and other times more closely related to bacterial homologs. Perhaps interest in fusion models arose because BLAST searches suggest that different eukaryotic coding sequences are sometimes more closely related to archaeal homologs and other times more closely related to bacterial homologs. Yes, that's true. That's a problem. These weak domain-specific affinities do not need to be understood and alternative explanations found. No, I'm sorry, These weak domain-specific affinities do need to be understood and alternative explanations found. However, in our view, they do not indicate that the eukaryote genome arose as a mosaic pieced together from archaeal and bacterial genomes. Whoa. Independent origins? You mean like, an orchard model like the creationists have? However, in our view, they do not indicate that the eukaryote genome arose as a mosaic pieced together from archaeal and bacterial genomes. This sounds a lot like the creationist model. It is an attractively simple idea that a primitive eukaryote took up the endosymbiont mitochondrion by phagocytosis. A unicellular raptor with a larger, more complex cell structure than that of present-day prokaryotes is envisioned as the host of the ancestral endosymbiont. This scenario, which is not contradicted by new data derived from comparative genomics and proteomics, is a suitable starting point for future work. Acquisition of genome sequences from free living eukaryotes among basal lineages is a high priority. And that's the end of the article. And they all lived happily ever after. So I hope you've enjoyed this talk. And just reflecting on this, this is a good segue into the talk that Dr. James Carter and I hope to have about eukaryotic evolution having to be pretty much miraculous in the way it does business, especially for intracellular transport of RNAs and proteins. So it is encouraging to see a paper like this that really tells it like it is that even if one assumes endosymbiotic events and also homology between some eukaryotic proteins with archaeal and bacterial proteins, that this is still not an explanation for what makes a eukaryote a eukaryote. Phylogenetic reconstructions are really not explanations. Evolutionary biologists seem to think when you ask how does something evolve, they just give you a phylogenetic reconstruction, which is not anything. It's just saying, oh, this looks like this thing, therefore it evolved. But that doesn't reconcile it with how this could happen in terms of physics, chemistry, and probability. Not one iota. It doesn't explain how we get these orthologs that are unique to eukaryotes, or these membrane-bound organelles, these signature cellular structures. So again, phylogenetic reconstructions are just worthless garbage pretending to be science. And I'm glad at least one paper is calling it out for what it really is. So with that, thank you for joining us and suffering through these two parts of this wonderful, wonderful paper on the irreducible nature of eukaryote cells. I love that word, the irreducible nature of eukaryote cells, irreducible. So thank you again for joining us. Take care until next time. God bless you.